Hello everyone, I am Dr. Vijay Lakshmi, Assistant Professor of Zoology at Sri Sharda College for Women, Salem. Welcome to this module. Today we will be exploring the fascinating yet complex world of cancer cells, understanding how they differ from normal cells. Cancer is a group of diseases characterized by uncontrolled growth and division of abnormal cells. Unlike normal cells, cancer cells bypass regulatory mechanisms, allowing them to proliferate without restraints. So in this presentations, we will explore the key traits, vitamin characteristics that make cancer cells unique and understanding how these characteristics contribute to tumor growth, metastasis, and resistance to treatment. By end of the session, you will gain a clear understanding of how cancer cells behave, why they are so dangerous, and how researchers are developing targeted therapies to combat them. Let's get started. Introduction to cancer cells. Let's begin by understanding how cancer develops. Cancer starts when normal cells undergo genetic mutations that disturb their natural life cycle. While normal cells divide in regulated manner, cancer cells bypass this controls and continue growing unchecked. They evade mechanisms designed to maintain balance in the body, such as programmed cell death apoptosis, and acquire the ability to spread to other parts of the body. So let's explore how this uncontrolled growth patterns and resistance to destructions make cancer life-threatening diseases. To understand this better, let's examine the fundamental characteristics that define cancer cells. Let's take a closer look at one of the most defining characteristics of cancer, uncontrolled cell divisions. Have you ever wondered how our body ensures that a cell grows and divides in controlled manner? Normally, a cell passes through multiple checkpoints in the cell life cycles, ensuring proper growth and DNA replications. Next, uncontrolled cell divisions. Let's imagine a traffic system where red lights suddenly stop working, cars keep moving, leading to traffic chaos. Cancer cells behave in similar way. Normally, cells follow a strict cycle with checkpoints, ensuring proper growth and repair. However, in cancer, these checkpoints fail, leading to rapid and uncontrolled cell divisions. This happens due to genetic mutations in key regulatory genes. Oncogenes act like an accelerated pedal, driving uncontrolled cell divisions. Genes such as RAS and MYC become hyperactive, pushing cells to divide excessively. Tumor suppressor genes like P53 functions as a brakes, preventing excessive divisions. When these brakes fail, the cell continues multiplying, forming tumors. As a result, cancer cells bypass the body natural regulatory mechanisms, allowing them to grow unchecked and form large destructive tumor masses. Understanding how these mutations drive cancer progression help us develop more effective treatment stages. Let's explore this process in more detail. Have you ever noticed how in crowded space people naturally maintain some level of order rather than piling on top of one another? Our body cells follow a similar principle called contact inhibitions. In normal tissues, cells stop dividing once they come in contact with other neighbors, ensuring an organized structures. But cancer cells do not follow this rule. Instead of stopping, they continue to grow and divide, piling up on top of each other. So this uncontrolled growth leads to tumor formations which appears as an irregular disorganized mass. A classic examples of this can be observed in laboratory cultures. A normal fibroblast cells form a single orderly layer, whereas cancerous fibroblast grows in chaotic dense clusters. So this loss of contact inhibitions is a major reason why tumors expand aggressively. Let's explore how this contributes to cancer progression. Let's imagine if a group of people in community suddenly decided to break all boundaries and move into different neighborhoods without permissions. That's similar to how cancer cells behave. Unlike normal cells that stay within their designated tissues, cancer cells gain the ability to invade nearby areas and even travel to distant part of the bodies. This process called metastasis makes cancer far more dangerous. Cancer cells breaks free from the primary tumors, enter into the bloodstream or lymphatic systems and settle in vital organs like liver, lungs, or bones forming secondary tumors. This spread is what makes cancer difficult to treat and is the primary cause of cancer-related deaths. For instance, lung cancer cells may migrate to the bones, causing additional complications. Understanding metastasis is crucial as it helps scientists develop targeted therapies to prevent cancer from spreading. Now, let's take a closer look at how cancer cells achieve this. Let's imagine if a faulty machine in a factory was never shut down. It would continue to cause damage. Normally, cells with errors are programmed to self-destruct through apoptosis. However, cancer cells disable this safety switch, ensuring their survival. They do this by mutating genes like P53, which normally trigger cell death when damage is detected. Additionally, they overproduce BCL2 protein that prevents apoptosis, allowing them to persist when they should be destroyed. So this resistance makes them particularly challenging to eliminate as many treatments aim to trigger apoptosis to shrink tumors. Next is the sustained angiogenesis. Just like acid needs roads to transport supplies, tumor needs blood vessels to fuel growth. So cancer cells achieve this by hijacking the body angiogenesis process, stimulating the formation of new blood vessels. They release signals like vascular endothelial growth factors to attack blood vessels, ensuring a constant supply of oxygen and nutrients. Unfortunately, these same vessels also act as a highways for cancer cells to spread through the bloodstream, aiding metastasis stage. Scientists are developing drugs like avastin to block this process and cut off the tumor supply lines. Now let's understand immortality. Normal cells have a limit on how many times they can divide. This is called the half-life limit. It happens because telomeres, productive caps on chromosomes, shorten with each division. But cancer cells activate telomerase, an enzyme that keeps telomeres long, allowing them to divide indefinitely. So this is why the cancer cells are often described as immortal. They don't age or die like normal cells. Let's understand genomic instability and mutations. We are all well known that cancer cells are like rule breakers in a genetic blueprint. They accumulate mutations rapidly due to defective DNA repair mechanisms. This instability allows errors to pile up unchecked, making cancer cells more aggressive and resistant to treatments. For instance, mutations in PRCA1 and PRCA2 genes significantly increase the risk of breast and ovarian cancers. Because cancer cells continuously evolve, they can adapt to treatments, requiring researchers to develop new stages to stay ahead and fight against the cancer. Let's see Warburg effect, that is alternative metabolism. So cells usually rely on oxygen to generate energy efficiently, much like how cars run based on high quality fuels. However, cancer cells shift to an inefficient method called Warburg effect. So where they rely on glycolysis even when oxygen is present. So this metabolic switch allows them to rapidly produce energy and essential biomolecules needed for their uncontrolled growth. So this abnormal metabolism is why PET scans which detect a high glucose consumptions are often used to locate tumors. Nowadays, the scientists are now exploring metabolic inhibitors as a potential way to stop cancer cells. As we know, our immune system is like the body's security team, constantly scanning for and eliminating abnormal cells. However, cancer cells have developed invisibility clocks to bad directions. So they do this by expressing PDL1 protein that deactivates immune attacks by switching off T cells. Additionally, some cancer cells mutate to hide from immune recognitions. New treatments such as immunotherapy drugs like checkpoints inhibitors are designed to block this evasive tactics, allowing the immune system to recognize and destroy cancer cells more efficiently. In summary, cancer cells possess several distinct and harmful traits that differentiate them from normal cells. This include uncontrolled division, invasion, and metastasis, resistance to apoptosis, sustained angiogenesis, limitless replications, genomic instability, altered metabolism, and immune evasions. Each of these characteristics add to this complexity of cancers, making treatment challenging. However, by identifying and understanding these hallmarks, researchers are developing targeted therapies designed to exploit cancer vulnerabilities and improve treatment outcomes. Through this session, I conclude that cancer research is constantly evolving with scientists exploring innovative ways to target its unique characteristics. Immunotherapy, gene therapy, and metabolic inhibitors are among the promising treatments being developed. Additionally, advancement in early detection and personalized medicine are improving survival rates. While there is still much to learn, ongoing research to paving the way for more precise and effective cancer treatments, offering hope for better outcomes in the future. By staying informed and supporting research, we all contribute to the fight against the cancers. So let's go through these possible questions and answer to enhance your understanding of the session. I hope this presentation has provided valuable insights into how cancer cell functions and why they are so challenging to treat. Thank you for your attention. We will meet again in the next module. Thank you.